disorder, this is a mouse where the MECP2 gene has been removed. And I'm just, going to play this video. This video is moving. You can see it shaking a little bit. This is a very sick mouse. If left to its own devices, this mouse would die within 48 hours. Okay? You can't really see it. It's not moving around the cage mouse. It's having breathing apneas. There's many things that are going on in this mouse. If you look in the brain, there's numerous problems with brain function. And in the second, they're going to pick up the mouse, and you can see its hind limbs clasped together, which is very characteristic of a neurodegenerative style disease, although there's no neurodegeneration in the brain of this animal. And as I said, if left to its own devices, this mouse is about 5 weeks old. This is the survival curve for these animals. You can see they're pretty much, they've all died by 16 weeks of age, but they really start dying at about 6 weeks of age. So what Adrian did was he just re-expressed the gene. And this was at 6 weeks of age, he re-expressed the gene in this mouse. Now 6 weeks of age I would not call an adult mouse, but it's moving into adulthood. And this is well beyond when the brain has typically reached maturation. So this is the exact same mouse, but 6 weeks later. Okay? And the mouse is walking around the cage. If you look in the brain, its synaptic physiology has returned to normal. Its breathing apneas are gone. It's lost all of the weight that it had because these animals tend to get very heavy. And in a second, they'll pick the mouse up by the tail, and you'll see that the motor coordination problems have been recovered, and they don't do the hind limb clasping, they just leave the legs splayed out. Okay? And if you look at the survival curve, once it's been re-expressed at 4 to 6 weeks of age, you can see this displaying, you can see that these animals no longer die. So he uses a genetic trick. So we can do a lot with genetics in the lab. Basically what he did was he created a mouse where there was a stop signal in the gene. So the mouse is developing without MECP2 being expressed. Then, using a clever genetic trick, he cut out the stop sequence. So the gene is still there, and it just started to be read. So at 6 weeks of age, we can't do this type of thing. We can do it in our mouse models. And to mimic what would happen if we could do gene therapy. So essentially, this is a mimic of gene therapy. So this changed the way people thought about neurodevelopmental disorders. It really made us start thinking, we need to know for each gene whether these are problems of neurodevelopment or problems of neural maintenance. And for the vast majority of the genes, we don't know. For Angelman syndrome, we know it's neurodevelopmental. Ipe Gesma, who works in Holland, he did the similar types of experiments for Angelman syndrome, which is a UBE3A gene, and he could not get recovery in the adult. Okay? So for Rett syndrome, it's probably a problem of neural maintenance. So a huge component of it is neural maintenance. But for Angelman syndrome, it's almost certainly mainly neurodevelopmental. And for SYNGAP, we don't know. There's a couple of papers coming out now sort of hinting one way or the other, but we really don't know the answer to it yet for SYNGAP. So we're busy making the animal models that will address this.
What needs to happen for you to say, I think it's developmental or maintenance?
So it's a great question because almost some aspects of these disorders will be neurodevelopmental and other ones might be neuromaintenance. So that's what I'm coming on to now. How do we phenotype our animals? How do we learn about our animals so that we have a battery of tests that when we re-express the SYNGAP gene, that we can say this is being rescued and this isn't.
Yeah.
And that's what takes so long with these things because you have to have a battery of things in your animal models in order to be able to do these types. So I'm going to show you a couple of things that we think we know enough about that it makes sense to make the models. So coming back to the question of convergence, what we try to do is we try to really understand the pathophysiology and then compare them across animal models. And this was done by Mark Baer's lab. He really pioneered this work on Fragile X syndrome with Emily Osterweil, who I poached from Mark Baer's lab at MIT and brought her here. So she's now here at Edinburgh, one of the key members of our team. But what they found was tuberous sclerosis and Fragile X syndrome were on this spectrum. And what you do is you identify a cellular process. So in this case, it was synaptic protein synthesis. And this is just neural performance, however you happen to be testing it through behavior or through brain physiology. And this is just a schematic where they show that tuberous sclerosis, the mouse model of tuberous sclerosis, had too little synaptic protein synthesis. We knew with Fragile X, the mouse models had too much protein synthesis. And what they did was they combined the two, they just mated the two mouse models together, and that restored protein synthesis to normal and many of the behaviors in the mouse models to normal. It was a proof of concept study. Right? Doesn't have direct clinical application, but it was a proof of concept that these two disorders might be sharing common features and a common pathophysiology. So this is one of the cases where if synaptic protein synthesis is a convergent mechanism that's leading to some of the problems in these disorders, that we can target protein synthesis either bumping it up a little bit or bumping it down through the same drug targets. So it was a proof of concept study. So we did the same thing with SYNGAP, and this was a paper that was published a little while ago. Here's just that measure of protein synthesis. And you can see that in Fragile X in red and the Fragile X model mice, you can see that the protein synthesis is up. When we measured protein synthesis in the SYNGAP to see if it belonged on this same axis, it was way up as well. So it seemed that SYNGAP, the neurons or the cells of the brain in SYNGAP have too much protein synthesis as well. So we went in to see if we could rescue that protein synthesis. And so we started with a variety of different drugs, all attacking one particular pathway, the RAS-MAP kinase pathway. And that's because the gap domain of SYNGAP is a negative regulator of RAS and bumps down this biochemical pathway. So we went in using a variety of different ways of bumping down this RAS-ERK pathway. And the one I want to highlight is lovastatin. So the reason why Emily Osterwile chose lovastatin was because it tweaks the RAS pathway, bumps down the ERK signaling, which we know leads to this basal protein synthesis alterations. And sure enough, we were able to rescue the protein synthesis with lovastatin and other things in the pathway that help with RAS, or that lead to the protein synthesis, so that by targeting RAS-ERK pathway. But what you really want to know then is, okay, that's great. At a biochemical level, it looks pretty good, right? But what we really want to know is these guys. What's happening in the neurons in the brain? What can we affect moving up the ladder? Can we affect the neuronal pathophysiology, and can we affect the circuits and ultimately the behavior? And that is what we're busy working on. I don't have any answers to you right now. I can tell you a little bit about the brain. And one of the things about the way these neurons work is they talk to each other electrically, and they're not static. So the connections between these neurons, which looks like this, so this is zooming in on one of those brain cells, you see all these hairy protrusions coming off of them. We call these dendritic spines. They're called spines. And these are the points of connectivity between two neurons. So it's how neurons in the brain set up circuits and talk to one another. And we know that by looking at this, we can record their physiology. We get these little electrical currents. Right? And the size of these currents, to give you an idea of the size of these currents, each one of these currents is measured in picoamps. They're about 5 to 10 picoamps. And to run a 60-watt bulb in your house, you need about 500 milliamps, or half an amp. So we're talking, these are, compared to an amp, these are 1 millionth, right? Actually, 1 billionth of an amp. Right? So they're tiny signals that we can measure. And these don't stay the same. The connections can change. How we learn every day is by changing. What's happening in the brain is we change these currents. So each synapse has, we say it's plastic, right? And we can measure that in the lab. And we know that this plasticity in these currents is altered in Fragile X as a result of these changes in basal protein synthesis. And that's just shown here. Black is a typical mouse. Red is a Fragile X mouse. We can see these synapses changing in the lab. And when we looked at SYNGAP, they looked exactly like the Fragile X. So again, there's this convergence of pathophysiology between the synapse and the Fragile X. And when we treated with lovastatin, it's not on this slide, but when we treated with lovastatin, we could rescue this. So we could not only rescue the basal protein synthesis, but we could rescue the alterations in synaptic plasticity that resulted from that. But we still are left with, can we rescue the circuit biology and can we rescue the behavior? And the other thing that I highlighted at the beginning that I wanted to point out is we're studying this one brain area, the hippocampus. Right? So it seems to work quite well. Lovastatin seems to work quite well in the hippocampus, but before we can say anything about lovastatin more generally, we really need to know about these other brain areas. So this is where I come back to. This was our model for many years. Before we had all these gene editing techniques, we went from gene to mouse to therapy to clinical trial. But now we don't have to do that anymore, right? We can learn a lot more before we ever think about going into clinical trial. So this is the model that we use. We go from gene to rat, mouse, and human, so we can use human stem cells, and we are always in touch with Andy about the clinical data that he's getting out from EEG, from the MRI studies, and I'll come back to those in a minute, why I think this is all incredibly important if we're going to try to translate our fundamental studies into clinical trials. So we have recently moved incredibly heavily towards the rat. SYNGAP was the second rat we ever made at the Patrick Wild Center. We now have two different models of SYNGAP, one where we completely lose one copy of SYNGAP, the other one where we've just selectively deleted the gap domain. So because we know that there's a range of different mutations in the SYNGAP gene that lead to SYNGAP haploinsufficiency. And so we're trying to mimic the main two types, either missense mutations in the gap domain or full loss of function mutations. So we have both of those models, but we're constantly comparing them to all of our other models. And all of these models are between Edinburgh and Bangalore, and we're constantly comparing between the two. I have a separate presentation where I have 150 slides from all the behavioral results from all of these models. And it tells you quite a few interesting things. So why a rat? Most people look at me and say, Peter, a rat is just a big mouse, isn't it? But actually, a rat is separated in evolution by 12 million years from a mouse. To give you some idea of that, the humans and chimpanzees are only 6 million, diverged 6 million years ago. Okay? So they're very different species. They have very different evolutionary pressures on their behavior. So what we're asking is a very simple question. How many of the things that we see in the mouse actually translate 12 million years to the rat? Because if they don't translate 12 million years to the rat, they're unlikely to translate 85 million years to the human. Okay? So these are very important cross-species validation studies that go on. The rats can also be used for a range of other things that mice aren't particularly good for. Rats have much bigger brains, so we can do MRI. Drug companies love them because their pharmacokinetics and dynamics are different. And they have much greater cognitive flexibility and much better. And for autism, they have much more social interactions similar to humans that mice don't. Mice are very standoffish and loners, whereas rats hunt in packs and work together in communities. So there's various things that the rats give us that make them a nice complementary model. Now, I talked about species differences. If you look at the SYNGAP mice that have been made until I talked to them aboard today, but the vast majority of the mice models that have been made, they don't have spontaneous seizures. And one of the things that Sally Till in my lab noticed not that long ago, actually, this is a cage full of the rats that we use. Half of them have a LAC1 SYNGAP gene, they're actually the gap domain deletion gene, and the other one's a wild type. And I just want you to pay attention to this animal right there. See that little head bob? He's having what we think is the equivalent of a little absent seizure.
Oh, yeah.
And these animals have a lot. You'll see one right now. He's standing up. He's going to have one, and it's going to make him actually fall backwards. Okay. And then he gets up and he's fine again. These are. We now know that they're seizures. And by the way, what I started to say and then finish my sentence was that the mice don't have these. So we only see these absence-like seizures in the rats. And when Sally started to quantify that, here's if you look at 18 animals that showed no seizures, 17 of them were wild type, and there was one SYNGAP heterozygous that didn't have it. If we looked at the 23 animals that did have seizures, that were showing these head bobbings, they were all SYNGAP heads. And we can now, that EEG, a rat EEG has been developed where we can have 32 electrodes on the skull and measure the EEG, and now we can show that these are actually seizures.
Can I just ask a possibly really stupid question? But do
No
they
such
have
thing.
seizure types? that
We're busy
correlate?
trying to figure that out. They do have seizure types. They have the absence. They have, we've seen a few tonic-clonic seizures.
Because with a drop seizure, obviously they don't have very far to go to the ground.
Yes. So I didn't put the pictures, I didn't put the videos in because I typically don't use them because they look pretty horrible. You can see there are rats that go into full tonic-clonic seizure. This is the main type we see. So most of the SYNGAP, if not all the SYNGAP hats, will show this type of seizure, this absence-like seizure. But we do see the full
You
tonic-clonic.
don't see the island one.
They just have these full tonic-clonic, where they lose all motor control stiffness. But very few of them show that. We're looking at ways that we can induce that to see what environmental signals and what signals we can do, whether through auditory stimulation, et cetera, might drive them more towards those seizures. But this is rare. For animal models to show seizures, the CDKL5 kids have 65 seizures a day, some of them. It's a massive seizure problem in the CDKL5. And the animal models show no evidence of seizures whatsoever. So, and that's more typical than atypical. So in terms of modeling the disorder, it sounds a bit odd to say I'm really happy our rats are having seizures, but in terms of modeling SYNGAP haploinsufficiency, this is something that many of the other disorders don't have. And this gives us a very clear phenotype that we can now go and try to rescue in these sort of preclinical trials. So one of the key things is how do we actually test all these different brain areas? Because I said the lovastatin seemed to work very well in the hippocampus, but that doesn't mean it's going to have a beneficial effect overall in the animal. So we set up a series of behavioral tasks that tap in to these various areas. I'm just going to show you one now that Sally Till in the lab has been working on, which is called the fear conditioning paradigm. And the fear conditioning paradigm taps into medial prefrontal cortex, it taps into areas called the amygdala, it taps into your primary sensory areas as well, visual or auditory, depending on how you run the task. And by looking at the fear conditioning task, we can start getting a hint of whether these other brain areas are showing alterations, and if they are, can we rescue those as well? I can't tell you whether we can rescue them, but I'm going to show you that we can see phenotypes. So back to that synaptic plasticity, if we start comparing synaptic plasticity in these areas that are known to be involved in fear conditioning, do they look like Fragile X? So again, the Fragile X rat models on the top, the SYNGAP rat models on the bottom. When we look at synaptic plasticity, you can see you get altered synaptic plasticity in these areas in both the Fragile X and the SYNGAP, and they look pretty similar. But when we actually look at the behavior, this is the fear conditioning. So what we do in fear conditioning, an animal gets used to a box, they're put in a chamber where a small shock is delivered at the same time they get a flashing light. They actually learn that the flashing light is predictive of the shock. So the next day, if you put them in a box and show the flashing light, they will freeze just to the flashing light because they have learned that a shock's coming. So you can see, you can watch the brain learning in action. These are very small shocks, so if you continue to play the light to them, within about 10 presentations, they no longer freeze. They no longer care. Right? So it's quite a light stimulation. You don't want the animals to actually start really changing their behavior. You just want to tap into how the brain normally learns. So if we do this in the Fragile X animals, this is the amount of freezing in that second phase that a wild type animal would do. And you can see 1, 2 presentation, and the third presentation, they're already starting to what we say extinguish the behavior. They already have relearned that the light doesn't predict a shock. Here's the Fragile X animals, right? They freeze much less. And this is believed that to be a problem in the amygdala, where the amygdala hasn't made the association. One of the key brain areas has not made the association. Interesting, although the amygdala and the medial prefrontal cortex have shown similar plasticity phenotypes, when Sally did this in the synapse, we saw the exact opposite. We saw something very different. Here, these are our two different models. So one lacks the gap domain, which is here. One is just a small change in the DNA here that leads to a complete absence of the protein product. You can see both of these show much more freezing, and they don't. And we've now carried this out. You can see that they start to extinguish, but they extinguish much later. So even though the areas that we know in the brain are showing similar differences in synaptic plasticity, the behavior is the opposite to Fragile X.
Can I just say,
Yeah.
sorry, me again. So could you translate that, like with my son, something will happen that will scare him like a noise, and then the next time, he's already anticipating it.
Yeah.
Is that what you're talking about?
It's very similar to that, yes. So he's learned that whatever was
So
causing that
The
noise.
sensory experience just goes straight there and it's stuck.
Yeah.
And it stays there.
Yeah. So and what we don't know is whether this is a problem in this primary sensory areas, or whether this is a problem in the amygdala, or if it's a problem in the medial prefrontal cortex. We don't know, but somewhere in that circuit, something's gone wrong that has made these animals unable to lose that memory, or unable to extinguish that memory. So we're busy. Sally's busy. Sally's at the back if anybody wants to talk further to her about it. She's busy trying to understand what's going wrong. And now we're going in with lovastatin because we have the seizures, we have the fear conditioning, we have motor coordination problems that we can test on the rotarod paradigm. So we're going in to see whether lovastatin actually has an effect in the rats, and we'll let us do all of these things. So one last word about lovastatin. We've just published what I think is an exciting paper, but then I'd say that because it came from my lab. This just came out about Fragile X, and what we showed was that if we gave lovastatin to Fragile X rats, Fragile X model rats, from 5 weeks of age to 9 weeks of age, or 4 weeks of age to 9 weeks of age, and then remove the lovastatin, all of the benefits in the cognitive function that we were testing remained out to 6 months of age. So even though we removed the lovastatin, we were able to maintain the cognitive functions, which we know involve a big brain circuit. They were maintained well out to 6 months of age. So this was the first study to show that removal of a drug in any of the neurodevelopmental disorders could have long-lasting beneficial effects. And of course, lovastatin is currently in clinical trials for Fragile X. There's 3 different clinical trials going on in the States. One of the big issues is all of the youngest person in any of those trials is 8 years of age. And the maximum age is 50. So the age range over which they're doing is not captured by what we did. We'd be looking more from about 2 years of age to 8 years of age over the time point that we're treating, right? Maybe 2 to 12. So I think it's always important to know what the scientists did and know what the clinical trials are doing, because they're not necessarily testing. We're hoping that lovastatin at later ages may help in Fragile X, but we don't know. We're doing those studies now. But if the clinical trials don't work, it might not be because the science isn't working. It's just that the science is done in a different way. That was one of the main problems with the MLR trials in Fragile X, which I'm sure Andy and I are happy to talk to you about.
Lovastatin, is that the medication, is that a drug?
Yeah, sorry, lovastatin is a commonly used statin for hypercholesterolemia. Right? So lovastatin it's not licensed in the UK because we tend to use simvastatin and a few of the other statins.
We'll talk about it a bit.
Yeah, Andy will talk about it more in terms of its clinical, but it's used widely in kids. That's why Emily went to lovastatin to tweak the ERK pathway, because she knew that lovastatin was already approved and had been used safely in kids. And I think you'll talk about the study on rosuvastatin.
Yes.
Yeah. So one of the key things that we want to do, which I haven't touched on at all, very briefly, my last slide. It's your work. I've talked a lot about this bit, but I haven't talked about how we get to human that much at all. And one of the things that we really lack, we can't do the types of studies I just told you about with shocks involved and taking brains out involved. We obviously cannot do that in humans. but we need a way to compare directly what we see in a rat or a mouse to what we see in a human. And so we've been working hard with Andy and Andrew to develop MRI in both Fragile X kids and adults with Fragile X to our rat models. Again, Sally's led the way on this, but what we found, I think, is really encouraging is that the resting state networks, so just the regions of the brain that are active in quiet wakefulness when you're just sitting around thinking, are very similar between the human and the rat. And actually, when we look at, I'm not going to go into any detail because it's my last slide and Andy's staring me down. If you look at the differences between a Fragile X human and a typical human, or a Fragile X model rat and a typical rat, the differences in the resting state networks are very similar. And this gave me great hope that we're studying the same thing. And we can directly, hopefully, give a drug to a rat, see how it affects the resting state, give it to the human, and hopefully see the same effects on the circuitry. Because knowing how we're translating through is going to be critical prior to actually doing any clinical trial. And where a lot of these monogenic disorders, getting into an MRI to get resting state might be very difficult. Remember the seizure, remember the EEG. So hopefully what we see in the EEGs will also translate through to the human EEGs. And for disorders where getting somebody into a scanner becomes incredibly problematic, hopefully we can do it with EEGs. So these are the two methodologies that we're using to really try to help translate through the clinic. And I'll stop now, Andy. Sorry.